Hey, what's going on? It is Monday, January 10th, 9:52 a.m. And today I'm going to talk to you about the immune system. What do you know? Again, I've been talking to you about the immune system for quite a while, and there's a lot to cover because I'm going to cover everything that's needed to understand the human immune system up to the point where the average postdoc understands it. But I'm getting to assemble all of these ideas and facts into my head, and I'm trying to put it all together. And it's long going, but consistent learning, consistent focus on the different pieces of the immune system make it easier to comprehend all the material and the several hundreds of different pieces that are involved. It's kind of like the brain, works by putting together little pieces and connections, which is difficult at first, but then later on connections are reinforced, learning occurs, etc., etc. Anyway, I've talked enough about that. So today I'm going to talk about the immune response. Okay, and specifically, I'm going to talk about the primary immune response and the secondary immune response. Okay, I'm also going to get into toll-like receptors a little bit, dendritic cells, and a few other pieces of the immune system that are kind of jointed and disjointed, and they're all related. Everything's really related, but we'll kind of weave them together as time goes on. So, the primary immune response. So basically, when you get infected by a pathogen for the first time, when your body gets infected by the pathogen for the first time, you have an innate immunity that initially kicks into gear right away, right? But your adaptive immune system, which works together with the complement system and the innate immune system, is still is not active, right? Because it's never seen this disease, at this point, and it's got no antibodies to help tag the pathogens and destroy the pathogens and help the cell-mediated immunity and the humoral immunity destroy and deal with the pathogens. So there has to be initial ramp-up to this, initial recognition situation going on. So what happens is first the pathogens enter your system with their associated antigens. Your antibody concentration is 0 at first, right? And this is basically your latent phase, right? Latent meaning hidden, right? And in the latent phase, there's a ramp up to antibody buildup. And recall how antibodies build up. You have your T cell interaction with your macrophage interaction and dendritic cell interaction, and then your T cells activate your B cells, which then produce antibodies through the creation of plasma cells, right? The plasma cells actually, with their heavy endoplasmic reticulums, are protein complex manufacturing factories. One of those complexes is the antibody. So the antibodies take time to build up in your system. So if you can see in this upper corner here, your primary immune response, which is the latent phase, is 0 antibody concentration, will then start kicking into gear your antibody production into the what's called the logarithmic phase. So that's the actual phase when accumulation starts to occur of antibodies in your system that are specific to the pathogen that has invaded your system. So that occurs for a while, and that occurs for about up to 2 weeks. Now it differs from and it makes sense. It differs from invasion of pathogen, infection to infection, right? But initially, this is the general response curve, right? So the logarithmic phase peaks at about 2 weeks, and in about 2 weeks, you have the most concentration of antibodies that you'll have, and then afterwards, there's after infection, at that point, the infection is presumably being dealt with maximally, and after that point, then the rate of infection, the rate of antibody production, and concentration in your system declines, right? Because then at that point, since the immune system is dealing with was dealing with the pathogen, so then you don't need as many antigens because presumably the disease isn't continuing in the case of an average situation, right? So you have some diseases which will continue for a long time, and that's a different story, and that's a more advanced immunology studies will require dissecting of that disease by disease. But let's imagine like a cold or whatever, and then so then your antibody concentration levels off, and your B cells are essentially your plasma cells are recycled. Your immune system then your B cells produce memory cells, right? So memory cells hold on to the specific antibodies and the specific genetic code through the somatic hypermutations that occurred during the course of the disease to deal with the next incarnation or the next time the disease occurs. So the next time the disease occurs, it's called your second exposure, right? And your second exposure to the antigen, as you notice in this curve, has the latent phase is again 0 because the antibody concentration is low to almost nothing, right? But then the response shoots up really quickly, right? Because at that point you have memory cells already. You have your hash tables, right, of antibodies that already are sensitive to the antigen that the new bacteria has produced, or new bacteria, infection, virus, is what would have you produces, and then the antibodies are produced faster, and the response is larger. That's something to note too, because essentially, the response can clear out once your body realizes that you're still in a situation where you can be exposed to the pathogens that you've dealt with in the past, so your body wants to clear them out faster because it can, essentially, right? And by doing that, the infection lasts a much shorter period of time. If you look at the peak occurs essentially a week earlier, right? The peak of the antibody concentration, which is the peak of your immune response, and then the immune response levels off. Okay, so but also notice that the response is faster, the response is larger, but notice that the response is also level, like the concentration of antibodies is much higher for much longer in your system, because your body really apparently wants to make sure the disease is gone. It's not taking a third chance because it knows that another exposure could occur, right? If one exposure occurs, okay, great. But if another exposure occurs, that means the source of that exposure is there, and then your body, again, doesn't want to take chances. So, that's basically how that works. And another thing to note is that your antibodies in your primary response, you've heard of immunoglobulin M, IgGM. So this is the IgGM is the antibody that's most present in the first immune response, right? So the first immune response has a pentamer of antibodies connected to each other through this IgGM, right? So this is basically 5 antibody top Y sections connected to each other without the stem. Well, the stem is here, right? And but it's like it's shared amongst several different antibodies, right? There's 5 different stem sections, and then you have 5 different variable regions, 5 different constant regions, right? And then you have this J chain connection. So these are the antibodies that are present mostly in the initial immune response, and then later on the antibodies during the secondary response and during subsequent responses, and after your infection has leveled off, are IgG, right? So IgG antibody is the second antibody, and again, this is like your standard antibody, right? This is the one that everybody knows about, and it, one can presumably, in a very layman sense, determine that this, is much smaller because it needs to be more mobile. It can penetrate through barriers more easily because it's smaller, and it can attach more nimbly to the different pathogens and whatnot. So that's basically what's going on with antibodies. This is a 10-minute video, so I'm not going to get to the dendritic cells or toll-like receptors, but those are pretty important because the dendritic cells are they're involved as in part of the first response of a pathogenic infection, right? And we talked about immune cell structure before, how they have both major histocompatibility complex 1 and MHC2 to expose their antigens. Dendritic cells are something I want to get into next time, and also the toll-like receptors, because the dendritic cells signal the rest of your immune system, and the signaling process occurs after toll-like receptors are exposed to the antigenic particles, right? So that's a whole different thing. Your cells have to transcribe these signaling molecules and send them in different directions. So yeah, we'll get to that next time. All right, that's it. Thanks a lot.